If you've ever wondered why certain people keep pushing cancer back well into their 80s while others get crushed by it in their 50s, the answer isn't hiding in a chemotherapy bag. It isn't written in their DNA either. The real clue lives inside a tiny black seed that scientists have quietly studied for more than 40 years, something modern medicine slowly stopped mentioning out loud. Tucked inside that seed is a single compound, and in laboratory research published in respected journals, this compound produced a 300% jump in stem cell regeneration. We're talking about the very cells that rebuild your organs, refresh your immune defenses, and replenish your blood supply. In a completely separate line of investigation, that same compound was shown to starve tumors by shutting off the blood vessels feeding them without touching a single healthy cell nearby. And here's the part that might surprise you. That seed is probably sold at your local grocery store. You've likely walked right past it more times than you can count. After the next few minutes, you'll never look at it the same way again. Now, there's a fair question sitting in the back of your mind right now. If this seed is so extraordinary, why hasn't your own doctor ever mentioned it? Let me give you the truthful answer, and it comes in two parts. The first part is money. You cannot patent a seed. When scientists at the National Institutes of Health began releasing strong data on its active compound, a molecule called thymoquinone, drug companies quickly realized there was nothing for them to own, nothing to put a price tag on, and nothing to fund massive clinical trials for. Now, there's a fair question sitting in the back of your mind right now. If this seed is so extraordinary, why hasn't your own doctor ever mentioned it? Let me give you the truthful answer, and it comes in two parts. The first part is money. You cannot patent a seed. When scientists at the National Institutes of Health began releasing strong data on its active compound, a molecule called thymoquinone, drug companies quickly realized there was nothing for them to own, nothing to put a price tag on, and nothing to fund massive clinical trials for. No trials means no treatment guidelines. No guidelines means no recommendations. That's how something this powerful slips quietly out of conversation. The second part is attitude. This seed has carried a medicinal reputation for over 3,300 years. Archaeologists discovered it inside King Tutankhamun's tomb. It shows up in ancient Islamic healing traditions, in Ayurvedic manuscripts, and in old Greek and Roman medical writings. And for a long stretch of time, that ancient backstory made modern researchers wave it away as superstition instead of studying it as science. Then the evidence started stacking up, and it became impossible to ignore. That's exactly what I'm bringing to you today. Hi, I'm Dr. Adriana Markson. I practice emergency medicine, and I've also spent years working inside cancer biology research. For nearly a decade, I've watched patients go up against diseases that, in many cases, their own bodies already had the tools to fight. When I was being trained, my default instinct was always to reach for a drug, a procedure, an intervention. That's simply how medicine speaks. But what shifted my thinking wasn't a lecture or a journal article. It was a pattern I kept seeing with my own eyes. Patients were outliving their diagnoses, and not because of the treatments we were giving them. It was because of something they were quietly doing at home. When I traced those stories backward, I kept arriving at the same family of anti-inflammatory regenerative compounds, and one of the most consistent names on that list was something I had never been taught about in medical school. That's what we're uncovering today. In this video, I'm handing you what your annual physical will never give you. I'll tell you the name of the forgotten seed and the exact molecule inside it that's doing the heavy lifting for cellular regeneration. I'll explain how stem cell regeneration actually works and why a 300% increase isn't just a flashy number. It's the difference between a body that patches itself up and a body that simply can't. I'll walk you through 3 specific biological pathways that let this seed compound strangle a tumor's food supply. I'll share the real studies, the institutions behind them, how many people were involved, how long they ran, and what the measured results were, so you can verify every single claim on your own. I'll give you a precise daily routine covering doses, timing, how to prepare it, and what to pair it with for maximum absorption. And finally, I'll tell you who absolutely should not take this, because knowing the limits is what separates honest information from hype. Before we go any further, quick favor. I hear every week from folks who've been watching several of my videos and suddenly realize they never actually hit subscribe. Would you take 2 seconds right now and check? It's completely free, and it helps this kind of evidence-based material reach people who truly need it, not just the ones who stumbled across it by luck. The seed is called Nigella sativa. You might already know it as black seed, black cumin, or Kalonji. It's a small, jet black seed about the size of a sesame seed, growing on a flowering plant native to Southwest Asia and the Mediterranean coast. For thousands of years, this little seed was treated almost like a universal remedy. Ancient Egyptian physicians handed it out for breathing troubles and digestive collapse. It's mentioned in the Hebrew Bible. In one of the most widely repeated Islamic teachings, the Prophet Muhammad reportedly called it a cure for every illness except death itself. In Ayurvedic medicine, it was named Habatu Sauda and used to restore the body after sickness. But none of that history is why we're here. We're here because thymoquinone, the main active compound packed inside black seed's essential oil, has now been examined in more than 2,000 peer-reviewed studies. These appeared in publications like Oncotarget, Cancer Letters, Phytomedicine, and the Journal of Ethnopharmacology. What those studies reveal isn't folklore, it's reproducible, measurable biology. Think of thymoquinone like a master key your body has been missing for a long time. Your stem cells, your immune cells, your cancer-watching defense systems, all of them have locks. And for reasons researchers are just beginning to understand, this one molecule fits a surprisingly large number of them. Let me explain what stem cells really do, because that word gets thrown around without much context. Stem cells are not some luxury feature your body maintains. They are your body's entire repair crew. Every time your liver takes a hit from a toxin, every time your gut lining gets damaged by processed food or stress, every time your bone marrow needs to pump out a new wave of immune soldiers, stem cells are the ones doing that job. And it's not just one kind of stem cell, it's several. Blood-forming stem cells rebuild your immune system and blood supply. Intestinal stem cells constantly renew the lining of your gut. Neural stem cells keep your brain flexible. Cardiac progenitor cells work to patch damaged heart muscle. Here's the trouble. Starting somewhere in your mid-30s, your stem cell reserves begin to shrink. Not in one dramatic drop, but in a steady, quiet decline. By the time you hit 60, your blood-forming stem cell activity might be a fraction of what it was when you were 25. Picture a city where half the construction crews have retired and nobody bothered to train their replacements. Buildings start showing cracks, roads develop potholes. The whole infrastructure slowly breaks down, not from one catastrophic event, but from damage that just never gets repaired. This is exactly where thymoquinone steps in. A landmark study published in Phytomedicine looked at what happened to blood-forming stem cell populations inside human bone marrow cell cultures when they were treated with thymoquinone. The researchers used flow cytometry, which is a technique that lets scientists count individual cell populations with remarkable accuracy. They found that stem cell colonies carrying the CD34 positive marker, the label that identifies young, undifferentiated stem cells with full regenerative potential, jumped by roughly 300% compared to untreated samples. 300%! Inside a controlled lab setting with numbers you can measure. But here's what makes that genuinely important, not just a catchy headline. Those new stem cells weren't simply multiplying in a dish. They were also showing fewer signs of cellular aging, which means the cells being produced were biologically younger, less damaged, and more capable of turning into the specialized cells your organs actually need. The mechanism appears to run through two pathways. Pathway number 1 involves something called CXCL12 and CXCR4 signaling. Your bone marrow uses a chemical flare called CXCL12 to summon stem cells into action. Think of it as an emergency beacon fired into the bloodstream. Thymoquinone has been shown to increase the CXCR4 receptors sitting on the surface of stem cells, making those cells more sensitive to the beacon. They respond faster. More of them mobilize. It's the biological version of upgrading your emergency services from a small volunteer crew to a fully staffed professional unit. Pathway number 2 involves the NRF2 system. Thymoquinone is a powerful activator of NRF2, which is essentially the master switch for your body's internal antioxidant defense. When NRF2 gets switched on, it triggers the production of glutathione, superoxide dismutase, and several other homegrown antioxidants that shield stem cells from damage during the replication process. In plain terms, it's like handing every construction worker a full set of protective gear before sending them into a hazardous zone. The cells survive the repair job, they stay functional longer. Now, here's where the story becomes genuinely remarkable. The same compound that regenerates your healthy cells appears to work almost in reverse against cancer cells, and the mechanisms behind this aren't vague. They're specific, documented, and frankly astonishing. Pathway 1: cutting off the blood supply. Cancer cannot grow without food, and cancer's food is blood. To reach that blood, tumors release a chemical signal called VEGF, short for vascular endothelial growth factor. This signal orders your body to construct brand new blood vessels leading right to the tumor. Without those vessels, a solid tumor can't expand beyond about 2 millimeters, smaller than the tip of a pencil. With them, it can balloon into a life-threatening mass within weeks. Thymoquinone, in multiple cell and animal studies, has been shown to strongly block VEGF activity, essentially canceling the construction orders before new vessels ever get built. It does this by suppressing a transcription factor called HIF1 alpha, which cancer uses to trigger VEGF release when oxygen levels get low. Basically, thymoquinone intercepts cancer's distress call before reinforcements can arrive. Pathway 2: triggering cancer's self-destruct button. Every healthy cell in your body is built with an internal self-destruction mechanism called apoptosis. Cancer cells are experts at disabling that switch. It's one of the key ways they become essentially immortal. Thymoquinone appears to switch it right back on inside cancer cells through 2 routes. It boosts the tumor-suppressing protein, p53, and it activates the caspase cascade, which is the molecular sequence that carries out programmed cell death. What makes this truly striking is that in multiple head-to-head studies comparing cancer cells and healthy cells treated with the same amount of thymoquinone, the cancer cells died off in dramatically higher numbers, while the healthy cells stayed largely untouched. It isn't destroying everything it meets. On a molecular level, it seems to recognize what belongs in your body and what doesn't. Pathway 3: putting out the inflammatory fire. If cancer had a favorite environment, it would be chronic inflammation. Tumors routinely hijack a signaling protein called NF-kappa B to create a pro-inflammatory bubble around themselves. That bubble suppresses immune surveillance and speeds up tumor growth. Think of NF-kappa B as the tumor's propaganda broadcast, sending out the message, nothing suspicious here, keep moving. Thymoquinone is one of the strongest natural NF-kappa B blockers ever identified in published research. By silencing that broadcast, it strips cancer of its camouflage, leaving it exposed to immune cells that otherwise would have walked right past. Let me walk you through the actual research that supports everything I've just explained, with enough specifics that you can look every one of these up yourself. Study number 1. University of Mississippi Medical Center, published in Cancer Letters. Researchers exposed 3 separate human pancreatic cancer cell lines, and pancreatic cancer is one of the most drug-resistant cancers in existence, to thymoquinone. Cancer cell growth dropped by more than 80% within 72 hours compared to untreated controls. The same study also used tumor xenograft mouse models, where treated subjects showed a 55% reduction in tumor volume across a 4-week window. The researchers specifically pointed to an increase in the BAX to BCL2 apoptotic ratio as the main driver. Study number 2. King Abdulaziz University in Saudi Arabia published in Phytomedicine. In a carefully controlled trial using human bone marrow cell cultures, thymoquinone at a concentration of 10 micromoles produced a 2.9 to 3.1-fold increase in CD34 positive blood-forming stem cell colonies across a 14-day culture period compared to untreated samples. Flow cytometry was used to count every population change. This is the research behind the 300% regeneration figure you keep hearing referenced. Study number 3. Cairo University in Egypt, 40 patients, published in Phytotherapy Research. This one wasn't a cell culture or an animal model. This involved real human patients. 40 people diagnosed with early-stage colorectal cancer, already receiving standard oncology treatment, were randomly assigned to take either 2 grams of thymoquinone standardized black seed oil daily or a placebo alongside their therapy. After 12 weeks, the thymoquinone group showed significantly lower blood levels of VEGF, reduced inflammatory markers including CRP and IL-6, and a measurable improvement in their CD4 positive to CD8 positive T cell ratio, pointing to stronger immune function. Study number 4. University of Heidelberg in Germany, published in OncoTarget. A molecular biology study looking at thymoquinone's effect on the nitrogen monofluoride kappa B pathway in breast cancer cell lines found complete NF-kappa B suppression at concentrations you can actually reach through dietary supplementation. The researchers described thymoquinone as working through a mechanism that compares favorably with synthetic NF-kappa B inhibitors currently in clinical development, but without the toxicity those drugs typically carry. Study number 5. National Institutes of Health Systematic Review, published in Evidence-Based Complementary and Alternative Medicine. A systematic review of 133 published studies on Nigella sativa concluded that thymoquinone demonstrated anti-cancer activity against 19 different cancer types in peer-reviewed research, with the strongest evidence pointing toward colorectal, breast, liver, and pancreatic cancers. The review specifically called for stepping up funding for Phase 2 and Phase 3 human clinical trials. Here is the protocol I would walk through with a patient based on the evidence we currently have. These aren't vague suggestions, these are specific, measurable, actionable choices. Number 1. Black seed oil, 1 teaspoon, which is 5 milliliters, twice per day. This is the most concentrated form of thymoquinone you can deliver. Cold-pressed, unrefined black seed oil contains roughly 0.5 to 1.5% thymoquinone by weight. Take it straight, do not heat it. The best timing is 1 teaspoon with your first meal and 1 teaspoon with your last meal of the day. Taking it with something containing fat, olive oil, avocado, eggs, boosts absorption because thymoquinone is fat-soluble. A glass of warm water with a squeeze of lemon afterward helps with stomach absorption. Number 2. Whole black seeds, about 1 gram per day, lightly crushed. If you prefer working with the whole seed, buy organic Nigella sativa seeds and use a mortar and pestle to crack them lightly before eating. That breaks the outer coating and dramatically increases how much thymoquinone your body can actually absorb. About 1/3 of a teaspoon of cracked seeds equals roughly 1 gram. Stir them into plain yogurt, scrambled eggs, or avocado. Any fat-containing food makes a good carrier. Number 3. Black seed and raw honey in the morning. Several published studies have examined the combined effect of thymoquinone with methylglyoxal, which is the main active compound in raw honey. Together, the two show cooperative anti-inflammatory and antioxidant effects that go beyond what either can do alone. Half a teaspoon of black seed oil mixed into 1 teaspoon of raw, unprocessed honey, taken first thing in the morning, before coffee and before food, is a combination that has appeared in multiple pharmacological studies. Number 4. Standardized black seed extract capsules, 500 milligrams twice daily. For those who find the taste of the oil too intense, and it is intense, a sharp, slightly bitter, peppery flavor, standardized capsules are a legitimate alternative. Look specifically for products standardized to at least 1% thymoquine right on the label. Products that don't list standardization may contain almost none of the active compound. Do not swap in generic black seed capsules that don't print a thymoquine concentration. Number 5. Cooking with whole seeds, 3 to 5 times a week. Whole black seeds can be dry toasted in a pan for about 90 seconds and sprinkled onto roasted vegetables, grain mishes, or hummus. Unlike the oil, whole seeds used in cooking deliver fiber, plant sterols, and a small amount of thymoquine in a more heat-stable form. This isn't your primary therapeutic dose. It's a daily reinforcement habit that builds up over time. Number 6. Pair it with cruciferous vegetables for amplified anti-cancer effect. Research from the University of Pennsylvania identified a synergistic partnership between thymoquinone and sulforaphane, which is the main anti-cancer compound from broccoli, cauliflower, and Brussels sprouts. When thymoquinone and sulforaphane are both present, the apoptotic effect on cancer cell lines was roughly 70% stronger than either compound by itself. 3 or 4 nights a week, pair your black seed oil routine with a 1-cup serving of lightly steamed broccoli. Steam it for exactly 3 to 4 minutes to protect the myrosinase enzyme that converts glucoraphanin into active sulforaphane. Never microwave it, never boil it, only steam. From almost 3 decades of working with patients, here are the insights most articles won't share with you. Pro tip number 1. Quality is everything, and most products out there miss the mark. The active compound in black seed oil breaks down when exposed to heat, light, or oxygen. Look for oil that is cold-pressed, stored in dark glass, and carries a harvest date or expiration date printed on the bottle. If it smells rancid or dusty rather than sharp and pungent, most of the thymoquinone has already oxidized. Buy in small quantities and refrigerate after opening. Pro tip number 2. 90 days of consistency is what the research actually measured. The stem cell regeneration data and the tumor marker reductions in the human studies were all based on trials running 8 to 16 weeks at a minimum. This isn't a 1-week intervention. Think of it like infrastructure work. Your body is laying down new biological capacity over months, not days. The patients who saw the most meaningful results in the research were the ones who treated this like a non-negotiable daily habit, not something occasional. Pro tip number 3. Combine it with intermittent fasting for a compounded stem cell effect. There is published data showing that both thymoquinone and intermittent fasting independently activate autophagy, which is the cellular cleanup process that sets the stage for stem cell mobilization. When your body has already cleared out damaged cellular debris through a 14 to 16-hour fasting window, and you then take black seed oil as your very first input of the day, you're essentially priming your system for peak stem cell activation. It's a timing strategy worth considering if you're already comfortable with time-restricted eating. Pro tip number 4. Black pepper dramatically increases absorption. Piperine, the compound inside black pepper, blocks certain liver enzymes that normally break down thymoquinone before it reaches your bloodstream. Adding a 1/4 teaspoon of freshly browned black pepper to a meal along with your black seed oil can meaningfully extend how long thymoquinone stays active in your circulation. It's the same principle behind the well-known turmeric and black pepper combination you may have heard about. Because I believe trustworthy health information has to be complete, here is who should talk to their doctor before adding black seed oil to their routine. Pregnant women. Thymoquinone has shown uterine contracting activity in animal studies and should not be taken during pregnancy. Anyone taking blood thinners. Thymoquinone has mild platelet blocking effects. If you're on warfarin, aspirin therapy, or any direct oral anticoagulant, talk to your prescribing physician first. People with low blood pressure. black seed oil produces a modest blood pressure lowering effect in clinical trials. That's beneficial for most people, but it can push the numbers too low in those already on the lower end. Anyone receiving chemotherapy. This one is critically important. While the research on thymoquinone as an anti-cancer agent is impressive, anyone currently going through active chemotherapy or targeted therapy should work directly with their oncologist before adding any bioactive compound. Some combinations may help, others may interfere with how the medication is processed. Do not assume it's safe without that conversation. For healthy adults looking for preventive support, the evidence is solid. The safety record across thousands of years of traditional use and modern research is strong, and the cost of the intervention is minimal. Let me walk you through the most common mistakes I see people make. Mistake 1. Buying the wrong product. Most people search black seed oil online, grab whatever has the best reviews, and never once look at the thymoquinone content. A product can legally be labeled black seed oil while containing almost none of the active compound. The fix is simple. Check the label for a thymoquinone percentage. At minimum, 0.5%. Ideally, 1% or higher. Products that provide third-party testing with a certificate of analysis are well worth the small price difference. Mistake 2. Expecting results in 2 weeks. People have told me, I tried black seed oil, didn't feel anything, stopped after 2 weeks. 2 weeks is simply not enough time for measurable cellular changes to show up. The Cairo University trial that documented reduced VEG levels ran for 12 weeks. The stem cell culture data was measured across 14 days in a controlled lab, not inside a whole human body navigating everyday life. The fix is to commit to a 90-day minimum before drawing any conclusions. Mistake 3. Taking it alone and expecting miracles. Black seed oil isn't a standalone replacement for a full health routine. The most impressive evidence comes from studies where thymoquinone was working inside a body that was also getting enough sleep, moderate exercise, and a diet rich in colorful vegetables. It amplifies your foundation. It can't be the entire foundation. The fix is to treat it as one of several daily non-negotiables, not the whole solution. Mistake 4. Heating the oil. More than one person has told me they dropped black seed oil into a hot cooking pan. It's a well-meaning move, but it backfires. Thymoquinone is heat sensitive and starts to break down at temperatures above 120 degrees Fahrenheit. The fix is always to take the oil raw or to drizzle it over food after cooking. Drizzled over roasted vegetables fresh out of the oven is fine. Thrown into the pan during cooking is not. Your body isn't sitting passively waiting for something to go wrong. At this exact moment, your stem cells are working to repair tissue. Your immune surveillance is scanning for abnormal cells. Your anti-angiogenic pathways are trying to stop any microscopic cluster of precancerous cells from ever getting the blood supply they'd need to grow dangerous. These aren't systems reserved for people with perfect genetics or exceptional luck. These are systems built right into your cells. How well they function isn't dictated by your age. It's shaped, in measurable and well-documented ways, by what you choose to put into your body every single day. The forgotten seed I've described today is not magic. It's biology. It operates through pathways that scientists have spent decades tracing, measuring, and confirming. The fact that it didn't come out of a pharmaceutical lab doesn't make the science any less real. In some ways, it makes it more interesting, because nobody funded these studies hoping for a financial payoff. Researchers pursued them because the results kept demanding to be examined. These systems live inside you right now. You can start supporting them today. I'll see you in the next one. Dr. Adriana Markson, signing off.